Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about regulation of gene expression. Okay. In the previous video lecture we already talked about the DNA transcription translation. Means in the previous video lecture we talked about the central dogma of the cell biology in detail, in prokaryotes as well as in eukaryotes. We talked about the transcription, we talked about the translation, we talked about the protein synthesis, it means each and everything in detail. If you miss these video lectures, so go back to my channel and watch this video lecture because it is very important. We also talked this protein splicing and RNA splicing as well. Okay. Before going to explain the genetic means the regulation of gene expression, you need to know about the central dogma of the cell biology. In the central dogma of the cell biology, we talked transcription, translation, and replication as well. But we didn't talk about that how actually it is regulated, means how actually it is controlled by a cell. So today in this video, we are going to talk about that. So what we are going to talk about, let me write, we are going to talk about regulation of gene expression. Okay, now what is regulation of gene expression? First of all, let me explain word by word. Regulation means to control. To control. Regulation means to control or switch on, switch off of the gene. And then what is genes? Gene is actually, these are the sequences which are present in a DNA that are used for the protein synthesis. Okay, these are what? These are actually sequences which are present in a DNA for the protein synthesis or for the product simply. And what is expression? Expression means to produce or to show. Means gene expression means that when the gene are ready for the functioning, after this gene, from this gene we get the DNA. Means we have a DNA from inside this means first of all we have a DNA, and inside the DNA we have a genes, and the genes are transcribed, which we get the RNA, and after the RNA what we get the protein. This is actually called as gene expression. Whenever the gene are processed, means whenever the gene perform their function, whenever the process occur, central dogma process occur, we call that gene expression. Okay. So regulation of gene expression means, let me explain. Regulation of gene expression means that whenever the gene are expressed, whenever the central dogma of the cell biology are occur inside a cell, so then it is controlled by a certain type of the protein. This protein could be a transcriptional factor, it could be some other protein and enzymes. Through this enzyme and protein, when the gene expression are under the control, we call that gene expression. Moreover, we can also say that turning on or off means the turning on and off of what? The turning on and off of the gene. When the gene are turned on by certain type of the protein, then the gene are expressed, means the central dogma of the cell biology occur, means the DNA, RNA, and after RNA we get the protein. Okay, this is so this is actually the turning on and the turning off of the gene expression, but through certain type of the enzymes or protein. Okay, so this is actually called as a regulation of the gene expression. Now how? Let me explain the process with a diagram. Let's suppose we have a DNA. Let's suppose this is a DNA. All of you better know that most of the DNA are in a double-stranded. Okay, DNA are most of the time it is double-stranded. Now if we have a double-stranded DNA, let's suppose this is a DNA of the prokaryotic cell. Although okay, leave the prokaryote and eukaryote. Let's suppose this is a simple DNA. We have a DNA. Okay, now from this DNA we get what? We get the RNA. Let's suppose this is what this is RNA, and from RNA we get what? We get the protein. Let's suppose this is what? This is the protein. So this process is actually called a central dogma of the cell biology. This is not the a complete process of the central dogma of the biology, but somehow we call that central dogma of the cell biology. Now what happened? Inside this DNA, we have a particular area which we call that genes. Genes are that portion where the specific information are present, through which we get the RNA and which we get the product. This is actually the main target of the cell, okay, which we get the protein. This is actually called as gene. Gene is that portion, okay. It have some particular type of the sequences which are used for the synthesis of the protein. Now, if this gene are controlled by certain type of the protein, let's suppose there are some type of the protein binded on it. Okay, these are what? These are the proteins or enzymes. When protein and enzymes are bound here, so then what it can do? It can enhance, or it can start the turning on of the gene. So when the gene will turn on, and then we get RNA, and from RNA we get the protein. This protein is actually protein, or we also call it enzyme as well. Okay, then we get protein or enzymes which are used by the cell. Okay, when the cell does not need any type of the protein or enzyme, let's suppose the cell do not need the protein or enzyme, so what happen? Again, the cell are continuously to produce protein, but the cell do not need these proteins. So then what happen? These enzymes and proteins which are formed, it will bind to the portion of the genes. It will bind to the portion of the genes, and when it bind to the portion of the gene, so then what happen? The gene will be switched off. So then the gene will be switched off or turned off. This is called as, so when it is turned off, so then the whole process will be halted or stopped. The whole process will be stopped. Now RNA is produced as well as protein is also not produced. So when these things are not produced, so then we call that turning off of the gene expression. This whole process is called as regulation of the gene expression. So I hope this video is helpful. If you like this video, so make sure to hit the like button, share this video, and also subscribe the channel, and stay tuned, because in the next video lecture we are going to talk about level of gene expression, and we are also going to talk about gene expression in prokaryote and eukaryote as well in detail. So thank you so much for watching that video.